In this video, we will talk about the motor system and take a deep dive into the cellular process that allows for muscle contraction. Now, we all know what it feels like for our muscles to contract, whether it be internally like our stomach wrenching or our biceps curling while working out. However, it is important to recognize the process is different for smooth and skeletal muscles. We will be talking about the process for skeletal muscles. The two main components of this process are protein filaments, myosin or thick filaments, and actin, thin filaments. These can both be found in a microfibril, which is an organelle in the muscle cell. To describe myosin, it is made up of 6 chains, 2 of which have the myosin head, which is key for muscle contraction. Actin filaments are made up of proteins that have myosin binding sites. On the actin filament, you have the tropomyosin, which is a long structure whose function is to block the myosin binding sites found on the individual actin proteins, and troponin, which is a protein that holds tropomyosin in place. Now let's talk about the process. When muscle contraction occurs, action potentials arrive at the axon terminal of the motor neuron. The motor neuron is a nerve cell that allows for impulses to travel from the spine to the muscle. Next, the voltage-gated calcium channel opens, allowing for calcium ions to move down the electrochemical gradient. The flowing of calcium releases acetylcholine, which is a neurotransmitter. Acetylcholine diffuses through the synaptic cleft and binds to the sarcolemma, or the plasma membrane of the muscle cell. Once acetylcholine binds to the sarcolemma, sodium ion channels open and potassium ion channels close, depolarizing the cell and eventually sending action potentials to the T-tubules. You may ask, what are T-tubules? T-tubules are the extension of the sarcolemma that wrap around the microfibrils in deeper or non-surface muscle tissue. Now that the action potentials have gotten to the T-tubules, the voltage-gated protein's other T-tubules change shape, allowing for calcium channels to be opened, and calcium can now flow into the cytosol of the muscle fibers. This is a process called excitation-contraction coupling. Now that the calcium is in the muscle cell, it can interact with myosin and actin, forming a cross bridge between the two filaments. Calcium binds to the troponin of the actin filament, forcing it to change shape. The change of shape in troponin means that it shifts tropomyosin, which allows for the myosin binding sites on the individual actin proteins to be exposed. The myosin head binds to the actin, changing ATP to ADP and phosphate. This binding pushes off phosphate. The release of phosphate forces the myosin head to bend and pull the actin filament. Remember, these two are bound together, and this is a process called the power stroke. The myosin head binds to another ATP, and once this occurs, the head of the myosin and the actin protein detaches, and the myosin head returns to its original shape and position. This process repeats until there is no more impulses being sent from the spinal cord. To understand it differently, once there are no more motor impulses sent from the spinal cord to the muscle cells, there is no longer a release of the neurotransmitter acetylcholine. The decrease in the release of the neurotransmitter will not open the calcium channels, and there will be no calcium to bind to the myosin to restart the process. This results in the opposite, which is muscle relaxation.